Thank you very much. I am very happy for the opportunity to present some results of the work I did together with Jesper Hinrich from the Technical University of Denmark and Alexandra Lelevich, who is a researcher at IFPEN in France. I'm a postdoc at the University of Copenhagen in the research group of Rasmus Bro, where we develop new algorithms and methods to analyze analytical data. Today I will talk about some new approaches for analyzing GCxGC-VUV data, focusing on resolving petrochemical groups. I want to start the presentation in reversed order with acknowledgements first, because what I will talk about would not have been possible without the initiative of Alexandra, who reached out to Rasmus Bro, my boss, and she asked him if he could help her analyzing a data set that she has acquired during her PhD. It was a purely scientific curiosity, and Rasmus was then so kind to forward her to Jesper and me, because he knew that we have been working with this type of data in the past. So thanks to both of them. And I hope I can show today that this approach, this all samples at once, shift-invariant trilinear decomposition, actually provides a simple and robust way to analyze GCxGC-VUV data, focusing on group-level analysis of petrochemicals. We haven't published anything yet, and we may have to reconsider the name of our method because it's not really handy. Anyways, there are two main arguments that we want to make about the method. The first one is that it's simple to implement, so we need a small number of tuning parameters, which is due to the data properties, and I will go more into detail about that. And the other one is that we can actually accurately estimate peak patterns, UV spectra, and peak areas, which is related to the model properties. And I will also go more into detail about that. But first I would like to show you a more general overview over the data analysis workflow that we have been developing. Everything starts here with a set of GCxGC-VUV measurements, and in this work we have used the plug-in module that Alexandra has developed during her PhD to do some basic pre-processing like smoothing operations and downsampling. And then we fed the data into our decomposition engine to get, as a result, resolved spectra, GCxGC landscapes, and peak areas, or peak volumes. And we had also the chance to use an in-house library at IFPEN to verify that these spectra actually describe very nicely different petrochemical groups. So in the end, we end up with a group-level quantitative analysis that is where actually the only tuning parameter is this number of component selection, but we are honored to also automate that. Okay, but let's have a look at the actual data. I mentioned that this is a data set that has been measured by Alexandra during her PhD. So we have 14 gas oil samples that have been processed in different ways. I'm not a domain expert on this, but down there you can see the paper where she described the data set more in detail. We have 24 measurements, so we have replicates for some of the samples. And the task is, as I said, to quantify, actually, I think semi-quantify is, that's a mistake here. So the goal is to quantify the different chemical groups that are present in these samples. Alexandra also already developed a really nice approach during her PhD called the template-based method, or I have called it here the template-based method. So we have like a grid of elution regions, and we sum up peak intensities to get to the quantitative estimates for the respective chemical groups. So actually she also benchmarked this approach against different other techniques shown here, and her results showed good agreement, so we consider this as a really strong benchmark for our method. Some of the limitations, though, are that it is sensitive to retention time shifts, so I have to know where my chemical groups elute, and if I have retention time shifts, then I need to adjust the grid to not sum up the wrong peaks. And another thing is that for resolving co-eluting species, I also need some a priori knowledge of what co-elutants do I have where. So our aim is to simplify that, to make it less manual and also a bit more unsupervised. So the method that we propose is conceptually very similar to the type of methods that Maria also yesterday talked about. It is along the lines of PARAFAC, multivariate curve resolution. And what is nice about the VUV data is that we actually have here the Lambert-Beers law that we can exploit. So essentially we have two kind of strategies that we could follow. We can either decompose the data sets individually into these factors, and each of the factors then describe the quantity, we also get the VUV spectra to assign the factors to a chemical group, and then we have the landscapes of this component family. Now we try to actually decompose sets of samples, like as a whole. And for that, we first concatenate multiple samples into this very large data set, and then we factorize it to get the relevant chemical information. All right, so the difference to also the pipeline that Maria showed yesterday is that we don't do a region of interest. We don't run a region of interest algorithm before to identify spots that we want to deconvolute, but we just deconvolute the whole thing. And those of you that are familiar with PARAFAC might be a bit skeptical, because running like a 100-component PARAFAC model is usually not a good idea, because you get problems with local minima, convergence, and it's a bit messy. So we also have been skeptical, and the condition is that we sort of get a good approximation of the raw data for a small number of these factors. So we checked the data, and specifically we looked at the singular value decurves. That's a fancy word for looking how many principal components do I need to explain my data set comprehensively, like up to a certain amount of explained variance. And here we could see that we actually have a really nice low-rank structure in the individual samples, but still also in the concatenated data set. So after seeing that we only need around 10 components or even less to explain more than 99.9% of the variance, we were a bit more optimistic that we could actually get something meaningful with a moderately sized PARAFAC model in terms of number of components. But there's still the problem with the shift. And Maria also, I have to reference back again, I'm sorry. But Maria yesterday also mentioned that we have this very strong trilinearity assumption in the PARAFAC model. And that's a problem, because here I just selected a very simple example. I took one of the gas oil samples and selected a peak here, which could be like a region of interest that we selected. And here we can see that we have this second dimension retention time shift due to the temperature programming. And if we now if we now fit a one-component PARAFAC model to that, then we can see that I hope you can see that the model, which is blue lines, and the raw data, which is orange lines, they don't perfectly match each other, and it gets more apparent when we look at the residuals, where we can see this very typical pattern of trying to model something with shift with a model that cannot handle shift. The problem is now that we have here actually a small co-eluting peak that we would like to resolve as well. But if we fit a two-component model to this data, then the second component just tries to explain the shift in this big peak better. So we don't get this small peak resolved. If instead we work with this shift-invariant model, then we can see we actually perfectly, we get a model that perfectly capture this shift, and in the residuals we see there is only this small peak left. If we fit a two-component model, then we resolve also this one. So these are like some references where we explain the background of the method. Of course, there's some math involved, and I won't go into details. I think the most important take-home is that we use the fast Fourier transform inside the algorithm to correct for the shift. And in the papers, we also show that it's significantly faster than PARAFAC2, which would be a good alternative to use if you have shifted data. And there are also extensions of this model where we actually allow for more flexibility aside from shift, also peak shape changes. And we can do that without losing the nice uniqueness properties that we want to have with PARAFAC. Then now let's get to something a bit more practical, the results. So we fitted a 7-component model to the 24 sample, the 24 measurement large data set from Alexander. And then we got 7 extracted UV spectra that you can see here in blue. And then we compared those against the in-house library that we got from IFPIN. And we could see that the extracted spectra actually very nicely matched to already known spectra in the library, which allowed us to simply cluster the different components into the relevant chemical groups. We can see here that we have two components describing monoaromatics across the samples and 2 components describing diaromatics across the samples. For the quantitative analysis, we group those together. And the first thing to evaluate the quantitative performance was to look at the replicated samples. We had some samples for which we had triplicates, and what we could see is that the peak areas that we get, they have a relative standard deviation of below 10% across the different groups, which I think is quite good. But you can tell me if it's not. The more important figure here is actually the comparison of our approach, abbreviated with SIT, to Alexandra's method. And here we can see that the quantitative results are in good agreement, with monoaromatics being the exception. So here we have to look into details for why the results doesn't match so nicely. Now, if you look at the axis, you can actually see that those numbers are not identical, and the reason for that is that in Alexandra's approach, she used relative response factors to reweight her results, and we didn't manage to do that before the talk. But it's a simple scaling, and I think if you have these correlation, then likely when we rescale it with the relative response factors, it's gonna be nice. Okay, so all in all, I think it's quite promising results for a method that is fairly easy to implement, because we only have to set the number of components. Nevertheless, there are some things we want to try out. So first of all, we want to run more models also on individual samples, just to see if we get even higher resolution, for instance, resolve sulfur-containing components, and more, if we can extract more sample-specific differences, so to say. And then we want to automate this number of component selection, because for now it's still a manual step. And then, of course, understand where the deviations in the monoaromatics come from. And I think finally it would be super nice if we could integrate this pipeline into an existing project, just to foster a bit open source development. And I really like the presentation of Maria. So maybe there is an opportunity to collaborate. And with that, thank you very much for your attention, and I'm happy to answer your questions.